Hello, my name is Fred Wittinghofer. I'm an emeritus group leader at the Max Planck Institute for Molecular Physiology in Dortmund, in Germany. And this is my second seminar. And sort of the first one, I introduced you to the molecular switches called GTP binding proteins or G proteins. And in this second part, I will more concentrate on a particular aspect of our research, which is dealing with the mechanism of GTPase reactions and how they lead to a number of different diseases. And I will obviously focus mostly on Ras-like proteins that I talked about in my first seminar. So again, just briefly, the cycle for these proteins, the mechanistic cycle, they come in two flavors, these proteins, in the GDP-bound and GTP-bound state. They have nucleotide bound very tightly. They need guanine nucleotide exchange factors to release GDP for GTP, because in the cell there is more GTP than GDP. That's why the protein becomes loaded with GTP once GDP is off. They have a downstream effect in the GTP-bound form, interacting with some partner proteins. And in order for the switch to be switched off again, you have the GTPase reaction, whereby the protein splits GTP into GDP and PI. And this reaction is very slow, and it's catalyzed by proteins called GTPase activating proteins, which will be obviously the major thing that we will be talking about today. So what we're talking about really is a really basic biochemical reaction, namely the hydrolysis of phosphoanhydrines and it's similar to the analysis of phosphoesters. For example, when you have phosphorylated proteins which are phosphorylated on threonine, serine, or tyrosine, you have a similar nucleophilic attack on the phosphate by water. And obviously, people have been talking about this reaction a lot, because it is a very slow reaction for the reason that phosphates are highly charged, highly negatively charged, and the approach of a nucleophile, for example, of water, which is partially negatively charged also, is very, very disfavored. And that's why this reaction is normally very slow. So even though the reaction delivers energy, it is very slow, because you have to overcome the very high activation energy, which depends on what I just told you about the negative charges. And the higher the activation, you might know from your biophysical courses that the higher the activation energy, the slower the reaction, because the reaction rate is directly proportional to the activation energy. So nature then uses enzymes to lower the transition state energy and thereby make the reaction faster. And there's an interesting article that I bring to the attention of my students all the time from Francis Westheimer, who wrote an article many years ago, why nature chooses phosphate, because chemists never use phosphate as a leaving group, while in the biology is a very frequent leaving group. So because of just this purpose, because of just what I've shown here, that the phosphoanhydride bond or the phosphoester bond is kinetically stable. In other words, you can have ATP in water and it stays, or GTP in water and it stays forever, or hydrolyzes very slowly, but thermodynamically it's in principally unstable. And if you use an enzyme that lowers the activation energy, you can make this reaction very fast. So that's an interesting article that I would recommend you to read. For example, that's why DNA is stable, and that's why ATP is such a wonderful source of energy, because it is in principle delivers energy, but it's stable in aqueous solution. So let's start then with, first of all, Ras and its gap-mediated GTPase reaction, because that is really the paradigm for many of the things that have been developed afterwards. So this is the molecule Ras. It's a ribbon representation of the structure of the G domain of Ras. And you see it's heart-shaped because obviously we like it very much. We solved the structure in Heidelberg when the city of Heidelberg has this advertisement, ich habe mein Herz in Heidelberg verloren, which means I lost my heart in Heidelberg. But the most important reason it's heart-shaped is because people call it the beating heart of signal transduction. So it's one of the most important sort of molecules that regulates important signal transduction processes like growth, differentiation, and sometimes even apoptosis. And you probably know a little bit about the signal transduction process, because every textbook has sort of this version. This is one of the paradigms of signal transduction chain, whereby, for example, a growth factor binds to the cell membrane and binds to its growth factor receptor called RTK, receptor tyrosine kinase, whereby this becomes phosphorylated. It recruits the exchange factor SOS, which then activates Ras to the GTP-bound form, and then now Ras interacts with the downstream component, which is RAF kinase, which is the starting kinase for what is called the MAP kinase module, where one kinase activates the next downstream kinase, which is called MAP kinase, activates MAP kinase, and that activates MAP kinase, which then goes into the nucleus and activates transcription. So this is a very simplified version of what Ras is actually doing, and this is the first one to be discovered, first signal transduction chain. But now it's probably becomes more and more complicated, and this is still a very simplified version, but it shows you already the major sort of thing about signal transduction via Ras, in that many upstream sort of components come and activate Ras. It's either tyrosine kinase receptors or G protein coupled receptor, T cell receptors, all of them can activate Ras. And then Ras can activate downstream many components, not just RAF kinase, but also a molecule called Ras-GAP, PI3 kinase, PLC epsilon, and others. And they sort of mediate a number of signal transduction sort of reactions, which sort of somehow are sort of integrated somewhere, let's say in the nucleus by a transcription factor, and initiate, when the threshold is right, when the number of sort of reactions is right, initiate the cellular response, which can be proliferation or differentiation or whatever. And I will not deal with any of the aspect of this, because I want to concentrate on the switch-off reaction. So the switch-off reaction is, again, the scheme. You have seen this now many times. But what happens is Ras is also an oncogene. An oncogene that has two types of mutation, either glycine 12 mutated to any other amino acids, or glutamine 61 mutated to any other amino acid. And what this does biochemically is it blocks the GAP-mediated GTPase reaction. So you can imagine what happens. You have blocked the return to the inactive state, and that's why you now accumulate Ras in the GTP-bound form. You don't need any of the upstream signaling anymore, because Ras is already in the GTP-bound state, and now it has an effect that is sort of not regulated anymore, and that's why it leads to cancer. Yeah, so these simple mutations, and that's why obviously as a structural biochemist, this is a very interesting project to ask the question, how can it that a simple point mutation has such dramatic consequences? And it's also obviously, since Ras is the most frequent oncogene, so about 25% of the people that come to the clinic diagnosed with a tumor, they have a Ras mutation, one of the ones that I've shown you. At least these are the most frequent ones. So obviously every drug company also are working on trying to inhibit the Ras pathway, Ras signaling, as a way of treating at least Ras-mediated cancers. And there are many approaches that one can think of. I just indicated a few here. For example, you could think of, for example, Ras is farnesylated at the C-terminal cysteine, and there's an enzyme called farnesyl transferase that mediates that reaction. There are farnesyl transferase inhibitors that are just in the clinic tested for the efficacy. But you could also think of maybe inhibiting the interaction with downstream effectors. This is, for example, a structure that we determined, the complex between Ras and RAF kinase, or part of RAF kinase at least. Or you can even think of, and that's what we are working on still, sort of our dream sort of approach is, if the basic sort of feature of oncogenic Ras is that it cannot hydrolyze GTP, can we maybe think about making small molecules that would induce GTP hydrolysis on oncogenic Ras? This is probably a dream project, and we still sort of work on it to make it feasible. But I will show you, though, in the course of my sort of seminar presentation, that sort of the reason we still think is possible is that the chemistry of it should not be so difficult. And I hope I convince you that you sort of go with me on that point in the end. So we're talking about here a nucleophilic attack of water on the GTP, on the gamma phosphate of GTP, mediated by Ras Gap, and it produces Ras, GDP, and PI. a simple biochemical reaction, but if it doesn't work, it leads to very drastic consequences, namely cancer. So first of all, and you have seen that picture also now a number of times, this is my introductory slide actually always, is that the two amino acids that are most frequently mutated, either of them, in oncogenic versions of Ras, they are very close to the active site. So you see here the nucleotide here, that is the gamma phosphate, and you see this is glutamine 61 and this is glycine 12, are very close to the active site. So obviously, as you would probably predict or thought before, is that they are somehow involved in the GTPase reaction. I will obviously show you what they actually do and why the mutation leads to a block in GTP hydrolysis. And you also can see here from a surface representation of the Ras active site that if this is GTP, so it sits in it's bound to the surface, and you see that the gamma phosphate is still approachable from the outside, and that will be important in the context of what I will be talking about. And you see also, I talked about last time about magnesium being important. If there is no magnesium around, you also have no GTP hydrolysis reaction. So, as always, in sort of a mammalian genome, there are not just one Ras gap, but rather 12 or 13. I indicated here a few of these, and you see they all contain one particular domain of about 330 residues. This is the Ras Gap domain, which by itself is able to initiate the fast GTP hydrolysis reaction. And you see that all of these proteins are composed of different domains. Some are similar, some are vastly different, and they obviously are regulating Ras in different contexts of the cell due to their different domains that they have in addition. And I will be talking about one that was the first Ras gap to be discovered by Frank McCormick, which is P120 gap. And I will be talking later on about NF1, which is another important element for tumor formation because it's a tumor suppressor gene. So first of all, and again to remind you, the intrinsic GTP hydrolysis is very slow, but if you add the GTPase activating protein, it's fast. So what I've been doing here is I take radioactive GTP, gamma labeled for example, and then I measure the production of PI over time. You see down there, this reaction, this is at room temperature, is almost negligible. There's almost no hydrolysis of normal Ras at room temperature without gap. And if you now add a particular amount of Ras gap, you see there is a very fast reaction, or much, much faster. And under limiting condition is actually about 10 to the 5-fold stimulation of that reaction. So that is the way we want to look at it. We want to analyze how gap mediates this fast GTP hydrolysis reaction. So sort of initially, obviously, you ask yourself, what could be the mechanism of hydrolysis and what is the step that is catalyzed by gap? So you could think of Ras-GTP being in principle a fast GTP hydrolysis enzyme, but it needs to come into a sort of GTPase competent state by an isomerization reaction. That is very slow and is catalyzed by gap. Or you could think that the actual cleavage reaction, going from GTP to GTP-PI, is very slow and is catalyzed by gap. Or you could even think that all of that is still very fast, but the release of PI to product is very slow, and that is catalyzed by gap. For example, if you remember maybe having heard about the hydrolysis of ATP on myosin, myosin is, for example, a very fast ATPase, but the PI release is very slow and needs to be catalyzed by actin. So in other words, what is the actual step that is catalyzed by gap in this particular case? And I should maybe already say that it is the cleavage reaction itself, which is the major point of attack by GTPase activating protein. And that only in the presence of gap the other reactions become at least partially limiting at least the PI release. And I'll show you that later on as well. And although I've shown you that before, I maybe repeat that again. So what we use a lot in our sort of studies, where we do fluorescent biochemistry with fluorescent nucleotide, we use sort of a MUNT or mGDP or GTP analog, where you have on the ribose a fluorescent reporter group. So this would be either deoxyribose or ribose, and on the ribose you have bound via this ester bond this the MUNT group, which is very sensitive to the environment that it sits in, and that's why it gives us always very beautiful structural changes, as I will show you in a minute. And we also used stop flow for measuring fast reactions, because remember, the GTPase reaction, which is slow in the absence of GAP, becomes very fast, and so in order to analyze it really in detail, we need to do stop flow kinetics. So what you, for example, do, you have RAS labeled with MUNT GTP, so with the fluorescent version of GTP, you have GAP, and you shoot them together into a cuvette, into fluorescent detection cuvette, and you have the stop flow up here in order to make sure that you put only a certain amount of liquid from these two syringes into your reaction chamber. So when we do that, when we shoot these two things together, we see that there is a fluorescent increase if you use RAS MUNT GTP and a decrease. The increase, again, is very fast. It means the two proteins make a complex, and then over time, the complex dissociates, because after hydrolysis, neurofibromin does not bind to RAS anymore. And all of this is over, as you can see, after 1 second. So very fast phosphor transfer reaction in the presence of saturating amount of GAP. And as a control, we use RAS bound to an analog, GPPNHP, where you see between the beta and the gamma phosphate, you have an NH group, which cannot be hydrolyzed anymore. And now you have also a very fast increase, which is complex formation, but no dissociation, because that cannot be hydrolyzed. So that is also the proof that the dissociation is due to GTP hydrolysis. So one may ask, in this reaction here, you may ask, what on GAP, which residues on GAP, obviously, are involved in mediating this fast GTP hydrolysis reaction, the 10 to the 5-fold stimulation of the GTPase reaction. And obviously, you were thinking of which residue, which amino acid could do the job. Or is it more than one amino acid? And as a good candidate, we were thinking of an arginine, because if you look at another G protein, G-alpha protein from the heterotrimeric G proteins, it is known that that consists of two domains. So this blue domain is again the G domain, and the red stuff here is a helical extra domain. But in that helical domain, you have an arginine, which sits right smack in the active site. And it has been shown that that arginine is important for the reaction. And that has been shown in a very old experiment, because there is sort of a bacterial pathogen called Vibrio cholera, which induces cholera. And what it does, it actually modifies this arginine on this G-alpha protein. You see, you have NAD, and the cholera toxin transfers ADP ribose onto an arginine of G-alpha, which is shown here. This is actually from the textbook here. So this is a very old reaction. It has been analyzed many, many years ago, and it showed when you do this reaction, when you block the GTPase reaction on G-alpha protein, there is no GTP hydrolysis anymore, and the protein is now producing cyclic AMP. It opens up channels, cyclic AMP-gated ion channels, and then you get all the symptoms of cholera. So again, the question, would that arginine be a good candidate? So we looked for conserved arginine in all the Ras Gaps that I've shown you, and indeed we found one, or we found several. Most of them didn't make any difference, but there was one arginine, when mutated, shows the following pattern in the reaction. So you again have, in the green version, this is the wild-type version, fast increase due to complex formation, fast decrease due to dissociation after GTP hydrolysis. And with these mutants here, arginine mutated to either lysine or alanine or anything you like to mutate it to, there is again an increase in the reaction, but then it stays up there, no hydrolysis whatsoever, or at least under those circumstances, up to a second there's no hydrolysis. Which told you already, obviously, that this arginine must be essential for the reaction. So far, so good. The biochemistry was clear, but obviously you don't really know what arginine is doing in the context. You think you have an idea. It may be going into the active site, but again, we have to wait for the structure to tell us really what it does. First of all, though, let me introduce you to another sort of important concept for analyzing phosphor transfer reactions, and that is using aluminum fluoride complexes. You may wonder why such an inorganic molecule would be sort of important, but it turns out that if you look at the nature of the transition state, so this would be GTP, being attacked by water, you have a transition state where the phosphate now make a trigonal flat thing where the leaving group oxygen, where the nucleophilic oxygen and the leaving group oxygen on the other side are the axial ligands of this penta-coordinate phosphate. And this transition state can either be very tight, which is then called associative, or very loose, depending on the distances here between phosphate and the two axial ligands here. And then you end up with, again, tetragonal phosphate after with PI-free phosphate. And it turns out that aluminium fluoride, which has sort of distances between aluminium fluoride very similar to between phosphate and oxygen, and also they are also highly electronegative, that this is a very good mimic of the transition state of the phosphor transfer reaction. The only problem was that while, for example, kinesin or myosin or many other phosphor transfer enzymes use aluminium fluoride as a mimic of the gamma phosphate in the transition state, Ras or Rho or all of these Ras-like proteins never showed that. Here you see that experiment. So you take Ras MUN GTP. It has a certain fluorescent emission spectrum with a maximum at around 440, and now you add the gap NF1. There's no change whatsoever to the spectrum, so nothing happens. But if you now add aluminium fluoride, you see that now you get a blue shift first of all, and then an increase in the absorption absorption. That means there is a trimeric complex between Ras, NF1, and aluminium fluoride, where the aluminium fluoride sits in the gamma phosphate binding site. And that was very instrumental. And by the way, if you now do, let's say, an oncogenic mutants, which we know does not hydrolyze GTP, and do the same experiment, you see in the presence of aluminium fluoride and Ras, there is no fluorescent change. And now you add NF1, there is no increase in fluorescence because you have a mutation Q61L that doesn't hydrolyze. You can also take a mutation of NF1, for example, with the arginine mutation, and again, there would be no change whatsoever. So in other words, what these experiments again tell us is that Ras is an incomplete phosphor transfer enzyme. It needs the presence of a gap in order to look like a phosphor transfer enzyme. And if there's any mutation that blocks the GTPase reaction, either on Ras or on NF1, we also get no aluminium fluoride complex. So obviously all of this is nice in terms of doing biochemistry, but what really mediates sort of the GTPase reaction, the fast one, can only finally be sort of verified by looking at the structure of the Ras and Ras-GAP complex. And this is shown here. So that was sort of the paradigm for analyzing this type of reaction. So you see, for example, in red you see Ras, and down in green is the Ras-GAP domains from P120 gap. And what you see then here, if you look very carefully, you see that there is a residue from gap going into the active site. So you see that when it comes back, you see that right there. There is an arginine residue pointing into the active site of Ras, and what it does is shown in the next slide. First of all, in this slide it shows that aluminium fluoride really is the flat triangle that sits between the leaving group and the nucleophilic water. So there is a mimic of the transition state. And you see then also what are the residues that really mediate fast GTP hydrolysis. So this would be the phosphate. So we think now, we take away aluminium fluoride and think what the real transition state would look like. You have the nucleophilic water, and you have a transition state where they are bound to the gamma phosphate, and the glutamine is fixing the water relative to the phosphate by an acceptor and a hydrogen bond donor interaction. And the arginine, what we call the arginine finger, is first of all stabilizing the position of that glutamine, and it also delivers its positive charges of the amidino group to neutralize charges on the gamma phosphate. So these are the two residues that are really crucial for the reaction. Glutamine 61 from Ras and the arginine finger from Ras-GAP. And this already explains, for example, why mutations of glutamine 61 in oncogenic Ras mess up the hydrolysis reaction, because you can imagine if you have, for example, a leucine or whatever here, it cannot do this type of interactions here. So any mutation of glutamine 61 is oncogenic because it is direct catalytic residue. So the structure also explains why mutations of glycine 12 are oncogenic, and that's also can be easily seen here on this slide. You have glycine 12, which is mutated to any residue, makes it an oncogene. And then you see aluminum fluoride being this flat triangle. You see glutamine 61 being there, and you see the argine finger down here. And all very, very close together, indicated by these blue dashed lines, which are indicating that this is almost van der Waals distance. And now if you mutate, for example, glycine to the smallest possible amino acid, which would be alanine, you see that it would immediately mess up all these residues in the active site. So for steric reason, there can be no other residue in the active site in the transition state except for glycine. So the message from all of this was obviously that there is an arginine that we call the argine finger that pulls the trigger on the GTPase reaction. Without it, it's very slow, and with it, it becomes 10 to the 5-fold faster. We used also, coming back to the question, what is now rate limiting in the whole process? So the main rate limiting step of intrinsic GTP hydrolysis is the very slow chemical step, the slow hydrolysis. But using a different technique, we now find out that there is another sort of step that becomes partially at least rate limiting, which is, I will show you that in a minute. So we use time-resolved Fourier transform infrared spectroscopy. And with that we can do sort of almost atomic resolution. We can look at really atoms in the active site at a millisecond time scale. So if you see a fluorescence, an infrared spectrum of a protein, which has an amide1 and amide2 bands, which is sort of not very structured sort of information, because it's sort of a mixture of information on alpha helix, beta sheets, and so on. But not a great detail can be taken from such a picture. But what we do is we observe for a reaction from, let's say, from a protein goes from A to B, we observe the difference spectrum, which at least on this absorption scale does not show much of a difference. But if you look in more detail, so you see the absorption there is 0.8 and the absorption down here is 0.02. So we see very small but very reproducible changes in the course of the reaction when A goes to B, and we see negative peaks. That means A goes away, and we see positive peaks if B comes out. So we can observe things that are lost and things that come up in the course of the reaction, and we can follow these by FTIR. And so if we do, and so first of all, we need to trigger the reaction at a particular time point in order to observe millisecond time scale sort of structural changes. And in order to do that, we use again a different analog, and the different analog is caged GTP, which was developed by Roger Goody, a colleague of mine in the institute. And so this caged GTP is blocked on the gamma phosphate by this what is called the cage group, so it doesn't allow hydrolysis. But now you can, with a flash of light, you can cleave that cage group off, and now you have Ras-GTP, which then hydrolyzes to GTP and MPI. If you do that with, let's say, Ras without gap, what you see is that you have you for example see absorbance. So any absorbance, as you might know from infrared, shows an atomic vibration in a bond. For example, you see vibrations for the alpha, the beta, the gamma, which decrease toward the end and become zero after 2 hours 5 minutes. So the different spectrum subtracts the end spectrum from the starting spectrum, and then you see only the changes that are occurring during the reaction. And so that is sort of quite normal. There is a single exponential decay when Ras hydrolyzes GTP. No big deal. But what is interesting is when you analyze the gap-mediated reaction, because now you don't see a single exponential decay, but you suddenly see intermediates appearing. You see peaks that come up and go down, and the most important one is indicated by this number 1113. This is the frequency for that particular change. So you have to analyze what is each band doing, does it belong to. So what we're doing, so we have developed techniques, or our colleagues in the University of Bochum with whom we collaborate, they have developed techniques to find out what is each sort of bandwidth, what is each frequent absorbance change, what is due to. For example, you find for GTP that there is absorbance change coming up with a rate constant K2. So K1 is the photoisomerization, K2 is one reaction, and K3 is the next one. And if you do that, you get an increase and a decrease with K3. Then there is an intermediate, which is coming up at 1113, comes up with K2 and decays with K3. There is free PI coming with K3. From all of that, we can obviously conclude that there is a PI intermediate with this absorption here, frequency 1113 centimeter to the minus 1, which appears with a rate constant K2 and decays with a rate constant K3. So in other words, the release of PI is now becoming visible. So we see hydrolysis when this PI peaks come up, and we see decay when it goes away. And you see this here, for example, in real time, so the absorption change with the rate constant K2. So you see here the scheme here, Ras, when it's in the on state, goes to the PI state, and you see there is a protein-bound PI comes up, and the PI band goes down in the course of the reaction. So this is repeated a number of times, and you also see that there is an arginine finger that is coming in and out of the reaction chamber. So we can follow not just protein bands, we can follow the phosphate bands at atomic resolution with on a millisecond time scale. And that tells us now, altogether, that is sort of the message from all of this, that while you have sort of a high activation energy for the intrinsic hydrolysis reaction of 92 kilocalories per mole, you now separate the reaction in two partial reactions which have a lower activation energy, and that's why it makes it so much faster. So you have the first activation energy for the cleavage reaction, which leads to Ras-GTP PI, and you have the second step where you have release of PI, and now you have the product. So that is sort of, again, a general theme in enzymology, that an enzyme catalyzed reaction lowers the activation energy, not just by lowering one reaction, but also by subdividing it in two partial steps, each of which has a similar activation, a different activation energy. And here you see that this activation energy is 59 kilocal per mole and 66, so it means this is a bit higher, and that's why this is a partially rate limiting step of the overall reaction. So let me now give you, so that was sort of Ras and how it leads to tumor formation. We analyzed in detail how this functions, even on a biophysical level. But now let me come back to sort of why certain GTPase reactions, when they don't work, how they lead to different types of diseases. Neurofibromatosis is one of them. I already showed you that neurofibromin, the gene product of the gene, is a Ras Gap. And there is a disease called type 1 neurofibromatosis, where people have sort of café au lait spots on the skin, sometimes small tumors on the skin, which can sometimes be rather large and very disfiguring, which are caused by mutation or deletion of neurofibromatosis gene. And for example, when we sort of worked on the mechanism of GTP hydrolysis by a GAP and NF1, a colleague from the Charité clinic in Berlin came to us and told us that he had a sort of a female patient which died at the age of 35, and she has 3 sons indicated up there, which also have the disease. And if we analyzed, or if he analyzed the blood of the patient and then the tumor itself, he found out that there is a mutation in the sequence of neurofibromin, where the arginine is mutated to a proline. You see that down there. Arginine is mutated to proline. And obviously, I would tell you that if it wasn't the catalytic arginine. So it turns out they have a mutation in the catalytic arginine, and when you now do the GTPase reaction that I've shown you before, you see that you have with normal Ras, this is the blue, with normal NF1, you have the blue curve here. It means an increase in fluorescence and a decrease, which means hydrolysis. And now if you take this mutation R to P, you have increase, which means complex formation, but no hydrolysis. And there is another patient mutation that we have analyzed in the meantime. Again, arginine to anything else, Q in this particular case, leads to a block of its ability to hydrolyze GTP on Ras. So mutation of the essential arginine leads to the disease neurofibromatosis. Also I should point out that there are many other mutations in neurofibromin which also cause the disease, which is phenotypically very, very different in many, many different patients. Let me introduce you now to a different system that is interesting both from a biochemical standpoint, but also from a standpoint of a different disease that I will come to at in due course. So what I will be talking about is a molecule called RAP, which obviously has a cognate Ras Gap. And RAP is a close homologue of Ras, and the name derives from the fact that it is highly homologous to Ras, because RAP stands for Ras proximate. Even though it was considered to be a close homologue of Ras, it does something completely different. It obviously has the same cycle between GDP and GTP, so it works as a molecular switch, but it does not have to do with proliferation as Ras or differentiation, but rather it is involved in integrin activation or platelet activation or other things. So the biology of RAP is completely different. But why the biochemistry is so interesting is that RAP is the only homologue or the only member of the Ras superfamily that doesn't have a glutamine in the position where glutamine 61 of Ras is involved in GTP hydrolysis. So it misses the residue that we thought was absolutely crucial for GTP hydrolysis, and here it's not there. And obviously the question is why that is so, and how does Ras Gap then work on this system, and how does it stimulate the reaction? So first of all, let me introduce you to the Ras-GAPs. There are indicated here 5 of them, but there are more in the human genome. But these 5 Ras-GAPs all contain a domain that is highly homologous, which is indicated here by the light blue and dark blue staining, where the light blue stuff is somewhat different to the dark blue, and I will explain that when we look at the structure. So again, the domain organization of all these 5 Ras-GAPs is somewhat different. That means they're probably doing their job in a different sort of context, in a different biological context. The reason is also interesting is that the dark blue homology region is also conserved in a protein called tuberin, which stands for a disease that I will be talking about in the end. So that's why it was also interesting to look at that reaction. And the third region is interesting to look at that reaction, is indicated in the next slide. But before doing that, let me show you first of all that we do again a stop flow fluorescent assay. We developed a system where we can look at this reaction in a biochemical way and analyzing mutations and the speed of reaction and so on. So here again we have RAP-GTP interacting with Ras-GAP. You get a quick fluorescent increase, which is due to complex formation, and a decrease due to GTP hydrolysis and dissociation of the product. And it's all over again after 1 second, whereas without Ras-GAP, the whole reaction would take hours. So in other words, we again a 10 to the 5 stimulation of the reaction. But the reason this is also an interesting system biochemically and mechanistically is that there are a number of conserved arginines in Ras-GAPs, and obviously we thought that one of them would be involved in providing an arginine finger to the system. In fact, we mutated all of them to alanine, and none of them has any dramatic effect on activity, as one can see here. So even the worst reaction is still close to 0.5 per second. So in other words, there is not a dramatic effect if you mutate any of the arginines, which makes it unlikely that there is an arginine finger involved in the reaction. So the 2 residues, a glutamine, intrinsic glutamine, and the arginine finger in trans, which in Ras and Rho and others make the important sort of catalysis, are not here in the system. That means the whole chemistry must be totally different. So we looked at the FTIR of the system again, and it shows basically the same features, even though the structures are somewhat different. The basic features in that there is an intermediate, a PI intermediate, whose decrease is rate limiting for the reaction. Although it's chemically somewhat different, as the different absorption spectrum show, it is indeed kinetically a most important intermediate. But what was interesting, and we found that in this particular case, in the Ras-GAP case, and it was also found in other systems in the meantime, is that the GTPase reaction is reversible, which sounds sort of crazy because it's a downhill reaction. If you take GDP and PI in the whole system, you would never ever create GTP. But what happens is, if you analyze the reaction by using, instead of normal water, O18 water, which is indicated by this black dot, you would expect that in the reaction you get hydrolysis and then you get GDP and PI, which has one phosphate labeled as O18 oxygen, one oxygen labeled as one O18 oxygen. But instead of getting a PI with one O18 oxygen, you get PI with two O18 oxygens, with three O18 oxygens, and with four O18 oxygens. As analyzed here by a mass spec, you see where you get where you get all these four products analyzed by mass spectroscopy. So how does that happen? So it happens because on the protein you have this sort of long-lived intermediate with GDP and PI sitting in the active site, and before going into product, they can also do a back reaction to make GTP with one oxygen now on the gamma phosphate, and now if it reacts again with O18 water, you get incorporation of a second O18 into the product, and if it happens again, a third one and a fourth one. So although the overall reaction is downhill, on the enzyme you get a backward reaction. And you can never get it once PI is released, because then the activation energy for the reverse reaction is too high. So we solved the structure of Ras-GAP also. This is a two-domain structure where one domain, which you see now to the left, is the catalytic domain, the dark blue region in the homology diagram that I showed you, and the light blue stuff is the dimerization domain, which is not important for catalysis, but is there to dimerize the protein for whatever reason that we really not know. So in analyzing the catalytic domain, obviously you ask yourself, what are the conserved residues and which of those play an important role in catalysis? And the purple helix here that I indicated here is the most highly conserved region. So it's likely that residues on this helix are somehow involved in catalysis. And indeed, you can mutate many of these and you see certain effects, but the most dramatic effect it happens when you mutate N290, so an asparagine, sitting on this catalytic helix. When you mutate that, you get the following result. So we call it, by the way, the arsen thumb, to make a difference to the arginine finger. So it's an arsen thumb, as you will see in a minute why we call it an arsen thumb. So first of all, if you now look at the mutation that I talked about, so if you take the N290A mutation and analyze the reaction by, again, this fluorescent stop flow assay, you see that while wild type makes a complex and then decays into product, the mutation here makes a complex. This is even, by the way, tighter than the wild type in the wild type case, but there's absolutely no hydrolysis. The reaction goes on and on. It stays up there. It never goes down. Whereas if you make a mutation like H287 to alanine, you see that there is no complex formation because the fluorescence stays down here. So that reaction is dead because they cannot bind, but that reaction is dead because we think this is the important catalytic residue. We think that from looking just at the biochemistry. Obviously, to know what it does, we need, again, to solve the structure. So which we did. This is the Ras-GAP complex. And you see here, the red and the green stuff is Ras-GAP, and the blue stuff is RAP, and you see GTP. But what you also see, if you look at in detail, that there is, again, something pointing from the red catalytic domain of Ras-GAP into the active site, and that is an asparagine. Obviously, it's the asparagine that I've talked about, which pokes into the active site of RAP. That's why we call it the arsen thumb in relation to the arginine finger and the other systems. And if you look in detail in what happens at the active site, and if you compare it to three other structures of Ras protein and their cognate gaps, taking RUN and RUN gap, Ras and Ras gap, and Rho and Rho gap, where Rho and Rho gap is from the Rittinger et al., and the other structures are from us, you see that these three structures here have a glutamine pointing towards the catalytic water, which attacks the gamma phosphate, which is indicated here, which is with the aluminum fluoride transition state, which mimics the gamma phosphate that is to be transferred. And the red structure here has, instead of the glutamine 61, has a threonine, which is pointing away from the active site, has nothing to do with catalysis. Instead, what you see is that the purple helix here inserts this asparagine into the active site, just exactly at the position where the others have the glutamine. So the difference is here, the three other structures have a glutamine, which is in cis, and Ras-GAP have an asparagine in trans, which does the same thing, namely stabilizing the catalytic water. And if you look at the surface of the protein, this is RAP surface shown sort of as in surface representation, and added to it is just the helix from Ras-GAP. It sits on the surface and inserts this asparagine into the active site. So the gamma phosphate peeks out of that hole, and all Ras-GAP really does is it has on the helix this arsen, it puts it into the active site, and that alone seems to be, at least in a chemical sense, responsible for stimulating the GTPase reaction by 10 to the 5-fold, because if you mutate that thing, it still binds all right, but there's absolutely no hydrolysis. So that makes us sort of think about, again, the feature of designing anti-RAS drugs. If all it takes is to insert such a residue into the active site, from a chemistry point of view, it should be doable, but we obviously have to develop molecules which bind in a correct way onto the surface, which is not so easy, and we are still thinking that we might be able to do it. And this is, again, coming back to that. So as an approach to anti-cancer drug target, let's find molecules that induce hydrolysis of oncogenic RAS. And from what we have observed with Ras-GAP, we have hope that it can be done. And the second reason, or the third reason actually, why sort of working with Ras-GAP system is again that it's connected to a disease called tuberous sclerosis, a benign tumors where people come and have hematomas in many organs. But the most obvious feature and what the sort of name comes from is that people have, when they do an NMR of the brain, they have these sclerotic tuberous sort of things in the brain that you see in the NMR of the brain. The people have mutations in two proteins called tuberin and hamartine, and tuberin, as I showed you before, has a high homology to Ras-GAP in this dark blue region. That means the catalytic region. And if you now look, for example, where patient mutations in tuberous sclerosis, in tuberin, where they are sort of occurring, and this is an alignment of different Ras-GAP sequences and the aligned with tuberin sequence, that you see that the most highly conserved region is again this thing here, the red stuff, where you have the catalytic helix. And one of the mutations in a patient with tuberous sclerosis, and many actually have that mutation, is on this asparagine that we know is the important catalytic residue. So this is another version of what we have seen before. An important catalytic residue is mutated in the disease tuberous sclerosis. So let me, in the last 5 minutes, just give you another disease, just to make sure that it's not just the RAS or RAP system, that there are many diseases where the inability to hydrolyze GTP lead to very many different diseases. So this is retinitis pigmentosa. People have pigments on the retina, that's what the name comes from. And they lose vision, they lose the peripheral vision. So this is a normal person seeing that building, and this is a patient with the disease that loses more and more progressively its peripheral vision and then loses complete vision after sort of the disease develops fully. And there are a number of genes mutated in retinitis pigmentosa that are called RP12X and whatever. And there is one form that is called RP2. It's an X-linked disease where people have mutations in a lot of point mutation in a protein. We determine the structure of the protein, which looks like this. This is the beta helix domain, and this is another domain. And you see many of the mutations that you then analyze. You find out that what you see is that most of the mutations determine or mess up probably the structure because they are inside the hydrophobic core of the protein, and you would imagine that they don't do anything to the catalysis or to the interaction of this protein. But there are a few mutations indicated here, for example, E138G or R11 180 to H, K, or C. So these residues point into solution. So you would think that they do something important for interaction with other protein or in something, that they are involved in what these proteins are normally doing. So we solved again the structure of the complex between AL3. So we knew that RP2 interacts with AL3, another Ras-like protein. It's called AL because it's an ARF-related protein. And we solved the structure of that. And so you see this stuff here would be the RP2 in purple. And you see in green would be AL3 in a GTP-bound form. And what you see then, if you again look very closely, you see that there is a residue from RP2 right here, which point into the active site of the other. So one didn't know what RP2 was doing, but when we solved the structure, we could immediately see that it smells like an arginine, like a gap, because it puts an arginine finger into the active site of the other protein. And if you look at the active site in detail, you see this would be GDP again. This is aluminium fluoride. This is glutamine from AL3, and these are three residues from RP2, Q116, R118, E138. And obviously, all of these residues are mutated in retinitis pigmentosa. And you can see that the arginine is doing the same thing that you have seen now many times before, and is stabilized by these other residues. And all three residues, when mutated, mess up the gap activity of RP2. So here, the structure really told us what the protein is doing. There was no idea about the function, and the structure told us exactly that this is a gap for R3. So let me now come to the conclusions from what I've been telling you about. That first of all, maybe conclusions about Ras itself, because it's the most important oncogene, is an incomplete enzyme, cannot hydrolyze GTP very fast. But there comes Ras Gap, which stabilizes switch 2 and the glutamine 61, which is the important structural catalytic element. And it also supplies an arginine finger into the active site. You have glun 61 mutation. Any mutation of glutamine 61 is an oncogene because it misses then the catalytic residue, the system. And you have glycine mutants, which are sterically compromised to do GTP hydrolysis. There is no way that the arginine finger can go into its proper position when there is a mutation of glycine 12. I also showed you that there is a strongly bound GTP-PI intermediate, and that PI release in the system becomes rate limiting. Whereas without GAP, the chemical cleavage reaction is the rate limiting step. The second conclusion, more sort of general to the Ras superfamily, Ras proteins are all incomplete enzymes. They all hydrolyze GTP very slowly. They have cognate gaps. So each of the subfamily proteins, and sometimes even proteins within the subfamily, have a specific cognate gap that are required for fast GTP hydrolysis, for catalysis. Some gaps supply an arginine finger that I have shown you now, many different examples, Ras and RUN and RO. RAP gaps deliver an arginine and a glin. Some gaps supply an arsensum. I showed you the example of Ras Gap, and tuberine probably does the same thing. PI release is very often rate limiting. And what is even more important, and that is my message for the whole talk, is that the perturbed GTPase reaction is involved in a number of diseases: cancer, neurofibromatosis, tuberous sclerosis, retinitis pigmentosa, and many more that I haven't talked about. Thank you for your attention. But I would, before I stop, let me first thank the people that have done the work. So the oldest story that I've told you about is that of Ras and Ras Gap, which was done by three postdocs in the lab, Reza Ahmadian, Klaus Schäfzeg, and Rohit Mittal. The Ras Gap story was done by the students, Oliver Daumke, Astrid Krämer, Pata Chakrabarti, and Andreas Grima. And RP2 story was done by Stefan Weltel and Karin Kühnel. And a lot of the movies that I've been showing you are done by Ingrid Vetter, which also runs my crystallography lab. and she was very, very helpful in almost all the projects. And we have on the FTIR, we have collaborations with our colleagues in the University of Bochum, which is Klaus Gerbert and Carsten Kötting. Thank you for your attention.